Hello everyone, welcome to ITLS Academy. The topic of today's lecture is rebuilding the nervous system with stem cell. We are going to learn how can we rebuild our nervous system if it is damaged anyhow, and rebuilding will be done with the help of stem cells. Let us see how all this will happen. Starting with chapter, we will see stem cells bring new strategies for developing replacement neurons. First point, just a decade ago, not a long time, neuroscience textbooks held that neurons in the adult human brain and spinal cord could not regenerate. This was thought just a decade ago, but now it is thought that it all is possible to regenerate the nervous system. Before a decade ago, it was thought that once dead, central nervous system were gone for good. Next point is, in the mid 1990s, neuroscientists learned that some parts of the adult human brain do in fact generate new neurons, at least under certain circumstances. Moreover, they found that new neurons arise from neural stem cells in the fetal as well as in the adult brain. Next point, the discovery of a regenerative capacity in the adult central nervous system holds out the promise that it may eventually be possible to repair damage from terrible degenerative diseases such as Parkinson's disease. Researchers are pursuing two fundamental strategies to exploit this discovery. One is to grow differentiated cells in a laboratory dish that are suitable for implantation into a patient by starting with undifferentiated neural cells. The other repair strategy relies on finding growth hormones and other trophic factors like growth factors, hormones, and other signaling molecules that help cells survive and grow. In this diagram, this is a picture shown is of a neuron. This whole structure is a neuron. As we know that neuron contains cell body and axon. This is the main part, cell body, and axon will form the neuron. Cell body have some branches. These branches are known as dendrites. These branchings arising from the cell body are known as dendrites. This is axon. This axon contains myelin sheath. Myelin sheath is a covering that covers the neuron. This covering is breaked at some point, and these points are known as node of Ranvier. These are node of Ranvier. Next comes at the end of axon, there are some points that are known as synapse. Synapse is a point that connects one neuron with the other neuron. From this synaptic point, one neuron connects with another neuron. Here, the second neuron has started. Some receptors are also present on the synapse point, and this contains post-synaptic membrane and synaptic membrane. I will going to tell you what these two terms mean. If this is synapse is a point at which two neurons join. So here one neuron ends and a second neuron starts from here. So the point where one neuron ends is the synaptic terminal, and the second one from where next neuron starts is known as postsynaptic membrane. These synaptic points have some chemicals, as these synapse this neuron will have some chemicals that are released to transfer information. That chemicals or that molecules are known as neurotransmitters. Neurotransmitter, as they transmit neurons. So and going from one neuron to another neuron, and bind receptors on the neuronal cell body and dendrites. Let us see. These are chemicals that are released by the one neuron, and these are received by the receptors present on the another neuron's cell body and dendrites. The neuron sends an electric signal down its axon to synaptic terminal. This is the direction of transfer of informations or signals, which in turn release neurotransmitters into the synapse. These neurotransmitters are released here that affect the following neuron. The brain neuron that die in Parkinson's disease. We are taking this disease as an example. This is a disease that is related with nervous system, and most of the researchers doing research on this disease. That's why we are taking it as an example here. Release the transmitter dopamine. Oligodendrocytes supply the axon with an insulating myelin sheath. This is myelin sheath, and this myelin sheath is formed by the oligodendrocytes. We have seen what is myelin sheath, but now you must know that this myelin sheath is formed by the oligodendrocytes. Coming to the next point, some multiple approaches for using stem cells in Parkinson's disease research. The point is, efforts to develop stem cell based therapies for Parkinson's disease provide a good example of research aimed at rebuilding the central nervous system. Next point, as is the case with other disorders. Both the cell implantation and the trophic factors strategies are under active development. Both approaches are promising. This is especially true of cell implantation, which involves using primary tissue transplanted directly from developing fetal brain tissue. Next point is the neuron that die in Parkinson's disease connect a structure in the brain called the substantia nigra to another structure called the striatum, composed of the caudate nucleus and the putamen. Putamen, we can say, is a neuronal pathway that degenerates in Parkinson's disease. These nigrostriatal neurons release the chemical transmitter dopamine onto their target neurons in the striatum. Here in this diagram, you can see the brain is shown here. Some parts of the brain like lateral ventricles, nigrostriatal neurons are shown. This is striatum and it includes caudate nucleus and putamen. Substantia nigra is also shown here. This is a neuronal pathway that degenerate in Parkinson's disease. For the explanation of image, we can take that these points. Signals that control body movements travel along neurons that project from the substantia nigra. These are movement travel to substantia nigra to the caudate nucleus. These informations are going from this point to this point. And putamen, you can see here informations how these informations are going to putamen and caudate nucleus. These both are collectively called as striatum, as you can see here. These nigrostriatal neurons release dopamine at their start in the striatum. In Parkinson's patients, dopamine neurons in the nigrostriatal pathway degenerate for unknown reasons. Here in this diagram, you can see it is started from the blastocyst that forms from the zygote. Zygote, you know, is a structure that is formed after the process of fertilization. If we talk about this diagram, so this picture is a schematic representation of processes involved in neural differentiation. During gastrulation, neurulation starts with the formation of the neuroectoderm. Here is the formation of neuroectoderm and neural tube. neural plate and subsequent formation of the neural tube also. During these events, migration of NPCs and neural crest cells takes place as cells migrate to their destination. Here you can see neural tube elongation has occurred. These cells start migrating. This is the migration of cell. After the migration, cells undergo differentiation, apoptosis and patterning. Once at their destination, newly born neurons extend new neurites and form synapses. You can see here cells start migrating. After migration, they start differentiating. Here comes differentiation. These can also go to apoptosis and patterning. As you know, apoptosis is a process of cell death. So, if things are not going in a proper manner and if there are any kind of damages or any kind of irregularity, then cell will ultimately go to apoptosis. This apoptosis is the natural cell death. So, after differentiation, these cells can proliferate. Proliferation is a process in which different types of cells are made. After differentiation, these cells can also form glial cells. Glial cells are myelinated. These help in the formation of myelin sheath, that is a part of the neuron that covers the axon. Let us see what happens after the proliferation. After proliferations, cells undergo patterning. Patterning is a process in which various patterns are formed. Pattern is an arrangement of cells. Here it is decided that how cells will arrange in a manner to perform their functions. In the process of proliferation, neurite outgrowth can also be occur. Neurite outgrowth, you can see if this is cell body, there is formation of dendrites and many other outgrowths. Except this, neurotransmitter turnover is also involved in proliferation process. Another event that also comes under proliferation is apoptosis. Apoptosis is a natural cell death. If things are not working properly, then cell will ultimately go to death, and this death is not due to any injury. This death is natural. That's why it is known as a natural cell death, or it is also known as programmed cell death. Next is synaptogenesis. Synaptogenesis is the process of formation of synapse, and this process is also included in the proliferation process. Proliferation process further helps in gaining the electrical activity. Electrical activity is an activity that helps in transfer of informations from one neuron to another neuron. Or we can say neurons send a signal from one neuron to another neuron. So this is the whole process that is involved in neural differentiation. This starts from blastocyst and ultimately goes to various cell types. This is very exciting that stem cells can repair neurons because it means we have a nearly limitless supply of neural stem cells. These neural stem cells can become the neurons and glial cells needed to repair the central nervous system. Specific proteins known as neural stem cells factor can be added to induced pluripotent stem cells, that is iPSCs, to turn them into neural stem cells or NSCs. Scientists now report how brain stem cells make the decision to transform into new nerve cells. Stem cells are true jacks of all traits of our bodies, as they can turn into the many different cell types of all organs. This allows the tissues such as muscles or even brain to renew and to heal after injury. Next point, we have a nearly limitless supply of neural stem cells which can become the neuron and glial cell. I think this paragraph is repeated, guys. You can take it into consideration as we have studied, and these lines are repeated lines. We have nearly limitless. This paragraph is repeated from this point, as we have already discussed, so we are not going to waste our time on these lines again. I am telling you a brief description that new neurons in the adult brain arise from slowly dividing cells that appear to be the remnant of the stem cells that existed during fetal brain development. Since some of these adult cells still retain the ability to generate both neurons and glia, they are referred to as adult neural stem cells. Here in this diagram, you can see how these stem cells are used in the process of brain repair. Let us see. These are embryonic stem cells. As we know that these embryonic stem cells are formed from the blastocyst. Blastocyst is a structure that is formed just before the implantation of zygote in the uterine wall. So these embryonic stem cells goes for the differentiation, and after differentiation, in the further process of differentiation, these forms neural identity genes. Neural identity genes are recognized. Further, these forms embryonic and adult neural stem cells. Now, these embryonic and adult stem cells have many fates. These are some uses shown in this diagram. On the other hand, these embryonic stem cell can go for further differentiation. These are neural differentiated genes. After the performance of these neural differentiated genes. These cells further go for differentiation and these becomes mature, that is mature nerve cells. And as we know that mature nerve cell, this is neuron, mature nerve cell is known as neuron. And we have already seen a detailed structure of neuron, what parts this neuron have. Like this has a cell body, these are dendrites, this part is axon, the covering of this axon is known as myelin sheath. This axon has synapse, these are synaptic lobes, and these are connections with from one neuron to another neuron. Apart from these neurons, the embryonic and adult stem cell, these are novel target for pharmaceutical research and development in directed cell production. These can be used for further production of neural cell. These can be stored, and as we know that process for storage of cells is known as cryopreservation. Cryopreservation is a technique in which storage is done at a very low temperature. Next use of these embryonic and adult neuronal stem cell is that these are unlimited cell source for transplantation. What does this means? Actually, this means that if any person is suffering from a disease that can be healed by the transplantation of these neuronal cells, neural stem cells, so that particular person can get these stored neural stem cell, and also he can get direct these cells for the transplantation. And after the transplantation, these neural stem cell will start forming mature nerve cells in the transplant's body. Ultimately, the main use of these both techniques is brain repair. These can be used for the repairing of brain on a broad scale, and this is an emerging technology in the field of science and development. In the future, these brain stem cells will cause a very useful work. So thank you very much guys for watching this session. Follow ITLS Academy on various social media platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. Guys, you can also subscribe our YouTube channel ITLS Academy, and do not forget to press the bell icon so that you can get notified whenever there is any upload. You can also get the app of ITLS Academy on the Google Play Store. So thank you very much again for watching. If you think that this video is helpful, then like it, share with your friends and also subscribe our YouTube channel. Thank you very much everyone.